Junk DNA by Valentina Esper and Daniel Baute. What is junk DNA? In genetics, junk DNA or non-coding DNA describes components of an organism's DNA sequences that do not encode for protein sequences. Junk DNA is 98% of DNA. However, scientists have determined that the term junk is not an appropriate name for this type of DNA. After several investigations, scientists have found that junk DNA is very useful.
Functions of junk DNA. Comparative genomics reveals that some regions of non-coding DNA are highly conserved. Some non-coding DNA sequences are genetic switches that do not encode proteins but do regulate when and where genes are expressed. Regulates genetic networks. Some specific sequences of non-coding DNA may be features essential to chromosome structure, centromere function, and homolog recognition in meiosis. Some non-coding DNA sequences determine how much of a particular protein gets generated.
Other sequences of non-coding DNA determine where transcription factors attach. Pseudogene sequences appear to accumulate mutations more rapidly than coding sequences due to loss of selective pressure.
DNA fraction of junk DNA. The amount of total genomics DNA varies widely between organisms, and the proportion of coding and non-coding DNA within these genomes varies greatly as well. More than 98% of the human genome does not encode protein sequences, including most sequences within introns and most intergenic DNA. While overall genome size and by extension the amount of non-coding DNA are correlated to organism complexity, there are many exceptions.
Types of junk DNA sequences. Non-coding functional RNA. Non-coding RNAs are functional RNA molecules that are not translated into protein. CIS regulatory elements. CIS regulatory elements are sequences that control the transcription of a gene.
Introns. Introns are non-coding sections of a gene, transcribed to precursor mRNA but ultimately removed by RNA splicing during the processing to major messenger RNA. Repeat sequences, transposons, and viral elements. Transposons and retrotransposons are mobile genetic elements. Telomeres. Telomeres are regions of repetitive DNA at the end of a chromosome, which provide protection from chromosomal deterioration.
Thank you for watching our presentation. We hope you like it and learn a lot. Thank you, Miss Sophie, for your help and support.